Hello. Okay. Good afternoon, ladies and gentlemen. Today's seminar will be starting right now. Thank you for attending today's seminar organized by celluTEIN Bioscience. I would like to introduce Mr. Brett, CEO of celluTEIN. For today's seminar, we are pleased to have Dr. Songwoo Kim, Dr. John Michael Gonzalez, and Dr. Seksak. And Dr. Seksom Atamangkun. Okay, let me introduce briefly about our speakers for today. For Dr. Songwoo Kim, Dr. Kim is currently a professor of nutrition at Carolina State University. He graduated with Doctor of Philosophy degree from the University of Illinois in 1999. Has published over 235 peer-reviewed papers, which has been cited over 18,100. For our second speaker, Dr. Gonzalez, he graduated with Bachelor of Science degree from Texas A&M University, Master of Science degree from Soros State University, Doctor of Philosophy degree from University of Florida. Dr. Gonzalez spent over 8 years at Kansas State University and 4 years at the University of Georgia. And the last one, Dr. Seksom Atamangkun. He graduated from Kasetsart University in Animal Science programs, Bachelor of Science and Master of Science in 1977 and 1981, respectively. He pursued his Doctor of Philosophy in Poultry Science at Oregon State University in 1992. His research has been involved in nutrition and management of monogastric animals rearing under tropical conditions. So Dr. Kim will begin his sessions with the title, Intestinal Health and Microbiota, the impacts of growth of nursery pigs right now.
Hey, good afternoon. It's my big honor and big pleasure to be here. And I'd like to thank PB Bioscience for the kind invitation to Thailand. It is winter in the US, but I'm enjoying the nice weather here. Okay. So today, I'd like to talk about maybe just 3 things here. First thing, what happens in the intestine of pigs. I'm talking about nursery pigs, so talking about baby pigs. And how does the intestinal environment can be influenced by diet, and then how all those together will influence the growth of nursery pigs. But when I talk about the intestine, I'll spend time talking about interaction between bacteria living in the intestine and the intestinal epithelium for their crosstalk. Okay. I'm sure everyone in this room will agree that weaning gives very big stress to the pigs, mainly those stressors coming from the diet or coming from the enteric diseases. So at the same time, you can see that since we are weaning pigs very early age, their intestine is not fully mature yet. So all those together, it brings a perfect storm to the pigs. So usually if you don't handle these problems correctly, they will not eat and they will not grow. So here I'd like to show you how important about understanding intestinal health is in swine nutrition. So we had a review paper here. Basically what we did was we tried to count number of papers published in pig nutrition. So using the PubMed, I typed growth in pigs, basically showing how many papers published in pig nutrition. So in earlier day, 1975, globally, there was about 900 papers published in pig nutrition. But as of today, 2022, there's over 1,300 papers published in pig nutrition. So that's really big increase. So when I was a student, somewhere around here, 1990s, at that time, I was told that in pig nutrition, we know everything. There are not much things to do anymore. But it was not true. You can see that there are a lot more research going on in swine nutrition now. And then I added additional keywords here, adding intestinal health and microbiota in addition to growth and pigs. And the number of papers published in that using those topics are shown in the black bar here. So in pig nutrition, we are not much really understanding pig intestinal health until around probably year 2002, 2005. But after that, you can see the big increase in the number of papers published in the area of intestinal health, intestinal bacteria in pig nutrition. So as of today, about 40% of all the papers published in pig nutrition somehow are talking about intestinal health or intestinal bacteria. So that's this is a big trend in pig nutrition, trying to understand the intestinal health. So that's what I'm going to talk about today. The first is about the bacteria. So we call microbiota, those are a group of bacteria, especially in the intestine. So when we say intestinal microbiota, what are we talking about? This is what I see as a nutritionist. If you look at the intestine, there are a group of bacteria stay with digesta in the lumen. So we call them luminal microbiota. There are another group of bacteria staying very close to the mucosa layer. So actually attached to mucin layer there. And we call them mucosa-associated microbiota or mucosal microbiota. So are they really different or not? If they're the same, why should we worry about talking about those two groups of bacteria? Okay, so if I talk a little more about those two different types of microbiota, for the luminal microbiota, they are attached or stay with the digesta. So what they do is they really utilize the nutrient in the digesta, mainly fiber or some part of proteins. So what they do is they actively produce metabolites. So you know the short chain fatty acids, those are a very good example. And they grow. Whereas those bacteria stay with the mucosal layer or mucin layer. Okay. They have a role of directly interacting with the host epithelium. But at the same time, they also utilize nutrient around the area, so they produce metabolites. So here in this figure, you can see that the bacteria very close to the epithelium, they have a direct interaction with the host animals here. And often you can hear about the fecal microbiota in many papers showing, okay, there's a changes in microbiota in the fecal samples. So those are fecal microbiota, but those are the one representing what happens in the large intestine. So I hope you can remember luminal microbiota, mucosa-associated microbiota, or fecal microbiota. So when you see the papers, you should be able to differentiate them. It is one paper I found in Nature, Mucosal Immunology. This is very nice review talking about how they are different. The conclusion from this review is saying that the mucosa-associated microbiota, they are the one directly interacting with the intestinal immune system. So they talk with the host animals. Okay? Compared with the luminal microbiota, they may do that indirectly through their metabolites. So if I'm a nutritionist trying to understand intestinal health of nursery pigs, if I want to look at the role of microbiota, I have to look at the mucosa-associated microbiota. Okay, we had a study a few years ago trying to compare, are they really different or are they just same? So we took samples from the jejunum, we took the digesta or mucosal layer, we identified what kind of bacteria are there. So here, at the phylum level, luminal microbiota or mucus-associated microbiota, when you look at their population, they are very different. So if you look at the bacteria staying with the digesta, they are mostly firmicutes, which means they are actually lactobacillus. In this case, this is where I'm talking about the nursery pigs. But those bacteria associated with mucosa, they are not lactobacillus. They are mainly proteobacteria or bacteroidates, which are very different from the lactobacillus. So good examples are Prevotella, and those are the bacteria like to utilize fiber. Okay? And also you can see the Campylobacteria attached to the mucosa as well. And the lactobacillus is one of them too. So their population is very different. Luminal microbiota and mucosal-associated microbiota, they are very different. So when you do research on intestinal microbiota, we have to really specify which one are you interested. And I had a chance to have a larger number of samples with about 900 samples. I'm showing you the exactly same thing. Those bacteria associated with mucosa or luminal microbiota. If you look at their population, those are relative abundance. They are very different. Again, mucosal microbiota, they are mainly proteobacteria or bacteroidates and some of firmicutes, whereas luminal microbiota are mainly firmicutes, which are lactobacillus. Okay? Very different. Okay? So from here, a very mini summary here. If you are trying to understand the role of intestinal bacteria on the intestinal health and also their interaction among diets and intestine, we have to look at the changes happening in the mucosa-associated microbiota rather than luminal microbiota. So that's a mini summary of this introduction. Now we can quickly move to the intestine here, still introduction. Okay. So when we talk about intestinal health of nursery pigs, well, what should we look at? So that's the question. So this is intestine of a nursery pigs, like 6 weeks old pigs. And you can see the nice stomach here and the long, small intestine and large intestine. But if you look at this picture in the middle, the major portion of small intestine is called jejunum, right after duodenum and right before the ileum. But the jejunum is the location where all the diet will come and being digested into nutrient, and nutrient will be absorbed. So this is the main area for the nursery pigs. When we talk about intestinal health, this is the main area of the concern. There are a lot of things happening in the jejunum, not like a large intestine. If you talk about intestinal health of the nursery pigs, I would not be interested in looking at the colon health, because for the young pigs, we don't expect too much things going on here yet, right? Okay, so what I'm thinking about is when a piglet, after weaning, they receive a diet. Those diets, even though it's very well nutritionally balanced, it will come with anti-nutritional compounds like glycine, beta-conglycine, or some oligosaccharides like raffinose. Also, the feed can have some allergens, and even it could have some pathogens or toxins from the pathogens, and they will go into the intestine, mainly the jejunum, and then now we will expect some immune reactions. Right? And then, in a sequence of changes in microbiota population, it comes with the inflammation, and that is related to the oxidative stress. And then now, after that, we will see the epithelial damages, and then got leaking, and that is related to impaired intestinal health and reduced nutrient utilization and impaired growth. So this is what I think is we will see in the nursery pigs after weaning when they receive the diets. So in order to understand what's happening, to understand the immune response here, we want to spend just a few minutes talking about immune system in the intestine. So this is maybe, as a nutritionist, I'm not used to looking at all these types of immune cells, but I'll try to explain. So there's a GALT, that is a gut-associated lymphoid tissue. That's the immune system. But in the case of small intestine, it is Peyer's patches. Okay? That's the main site for the small intestine where all the immune cells are together. So there are different types of immune cells making Peyer's patch. One is the microfold cell. In short, we call them M cell. So hope you remember that M cell. Also, there are 3 types of antigen-presenting cells. One of them is dendritic cell. Another one is a macrophage and the B cell. Those are three types of immune cells which will take antigens from external environment and they will initiate the immune response. And then there are T cells waiting for those antigens. So that can be helper T cell or cytotoxic T cells. So all these immune cells are making the Peyer's patches. Okay. To make it easier, I spent a few hours drawing this. Okay. So these are the layer of intestine. You can see that this intestinal epithelium in the jejunum. On your left side, this is external side. It's called lumen. And your right side, that's the inside of the intestine. And here, the cells looking with the like finger type structure here, those are enterocyte, making the intestinal epithelium. In the middle, you can see a different kind of cell, like goblet cell. The function of this is to produce mucin. Okay? And there's an M cell, microfold cell. That is the cell which will try to sample the antigens from the lumen. Okay? So now we have antigens. So it can be part of bacteria, or some bacteria cell wall will have toxins, or some bacteria will actually bring some endotoxins, such as lipopolysaccharide, I think you heard about it, or lipopeptides, or what is that? I forgot. Anyone? Peptidoglycans. Okay. So all those antigens or the compound originated from bacteria will be taken by those epithelium. Okay? One is an M cell. M cell will take antigen to inside of the body, and LPS or peptidoglycan and others will be recognized by the enterocyte. And there are different types of receptors on the enterocytes. Okay? So in the body, now there are three types of cells are waiting. Dendritic cell, macrophage, or B cell, they're waiting. They'll recognize this antigen. Okay? And then they will show up, they'll show those antigens on their hands. Okay? And those antigens will be recognized by T cells after that. Right? So T cells will be activated by these antigen-presenting cells into helper T cell or cytotoxic T cells. So they are active T cells now. Also one T cell, like helper T cell, they can further activate B cells into plasma cell, and the plasma cell will produce antibodies against these original antigens. So this is just brief overview of intestinal immune system that we'll utilize this information in the next slide. So I'm showing you these immune cells again, dendritic cell, macrophage, and B cell, and T cell, and enterocytes. When they're activated, they will secrete signaling molecules, and you heard about them, which are called cytokines. So many different types of cytokines are secreted under this immune activation, but those are commonly have a common function of causing inflammation. So as an outcome of the secretion of these kind of cytokines, we will see increased inflammatory response. So intestine will have inflammation. When those immune cells are activated, also they are consuming a lot of oxygen and presenting or producing free radicals or oxygen, reactive oxygen species. So at the same time, when those immune cells are activated, they will cause the oxidative damages to the pathogens, but at the same time to the host animals too. Basically, intestine will be damaged by oxidative stress here. So as an outcome of activation of intestinal immune system, we have increased inflammation and we have increased oxidative damages. So that happens in the intestine of nursery pigs. Right? I'm going back here. So same picture here. So we have antigens or compound from the bacteria, and we have immune cells and the cytokines. They'll cause the inflammation, and also after inflammation, you see the oxidative stress, and then they will damage the host intestine. So there's epithelial damages. So those are all related together. So when pigs are weaned and they are getting the diet, comes with all antigens or even bacteria, immune systems are activated, and then you have epithelial damages. So as a picture here, so this is a picture of jejunum here with a very nicely developed villi. And after all this happened, you can see the destruction of villi here. So after that, what happened is the intestine will try to be rebuilt. So the cells in the crypt, they will start to proliferate again to build this villi again. So that's going on to the pigs after weaning. Okay, now I'd like to fit the bacteria, microbiota to this picture. So this is the same thing that we talked about. Now there's a mucus layer that we should not ignore. So mucus, the mucins are produced by these goblet cells. And now I'm going to put the bacteria. Those are, so they are attached to mucosal layer here. So I'll say the mucosa-associated microbiota here. Those are bacteria that we have. So those are bacteria, could be Prevotalaceae or even Campylobacter or Lactobacillus. Those are the bacteria we have all the time for the healthy animals. And someone can provide some probiotics, some like lactic acid producing bacteria as probiotics, or some other gram-positive bacteria. They are staying there all the time under healthy condition, and this M cell will actively check what kind of bacteria we have. And then also those receptors on the enterocyte, they will take some samples from those bacteria saying, okay, this is a normal condition. We don't have to fight against them because they're the one we have all the time. So there's no immune activation, which means there's no inflammation going on here. So this is a healthy condition. However, when there are invasions from the outside, like pathogens, okay, or other gram-negative bacteria, then those are unusual bacteria, and this M cell will take samples from that. Okay, so antigens are going inside of the body, and they will activate those antigen-presenting cells. But also, those enterocytes will take those samples from the pathogens, and those receptors will be activated. And then, after that, cytokines causing the inflammation will be secreted, and now you have activation of the immune system, inflammation, and oxidative damages, and epithelial damages. So that's what's going to happen when the piglets are challenged from the external environment. Okay. So I'm going back here. I'm now showing you the enterocyte here. Those are cells making the intestinal epithelium. So I'm showing you those receptors. Okay. Now I put the names here already also. Those are kind of new, but TLR is called a toll-like receptor, and NLR is called node-like receptor. There are different names of receptors, which will take samples from the external environment. So when under the healthy conditions with the commensal microbiota, normal microbiota, these receptors will kind of come down, will not cause inflammation. Okay? So it's a just normal condition. However, when there are invasions or some external challenges comes, and those receptors can be activated and causing the inflammatory response. Okay? So this could happen when piglets get challenged from outside. Okay? At the same time, I'd like to point out here, there's a enzyme called mTOR. So related to that, we call the mTOR signaling system here within a cell. When this mTOR is activated, will try to build a tissue or make a protein, or if this is a stem cell, it will try to proliferate. So under normal condition, this mTOR will not be activated, it's not needed to be activated, so it will come down like this. However, when these cells are challenged, immune challenges from external environment, these receptors are activated and causing inflammation. They will activate mTOR system also, signaling pathway too. And then if this is a stem cell in the crypt, this cell will try to proliferate to build the villi again. So that's the kind of overview of what happens as an outcome of immune challenge, normal or challenges situation. But then here, I'd like to make a list of different factors activating mTOR, different kind of factors inactivating mTOR. Because mTOR, activation of mTOR in the intestine, that's related to the repairing of intestinal tissue. So these are like amino acid, glutamine and leucine. Those are amino acid. They are known to activate the mTOR. And some hormones, anabolic hormones like IGF and insulin, even testosterone, are known to activate mTOR. Like a cytokine, pro-inflammatory cytokine, like IL-6, or even TNF-alpha, they are known to activate mTOR. However, they will be activating mTOR through activating inflammation. So normally, when mTOR is activated in the intestinal tissue, it comes with inflammation and oxidative stress, and mTOR is activated to rebuild the tissue. Here, these are the factors inactivating mTOR. So those are when piglets are not eating, so nutrient restriction will inactivate mTOR. Some other hormones like cortisol, they will inactivate mTOR, so that's a stress environment. And omega-3 fatty acid is well known. They have a well-known function of reducing inflammation, so they are anti-inflammatory. So omega-3 fatty acid is shown to inactivate mTOR too. Also, like antioxidants, very good antioxidant like gallic acid, that is also known to inactivate mTOR. So these are something that we see is a good thing. Good functional nutrients are inactivating mTOR. Okay? So those are two different situations. But here, trying to enhance the intestinal health for the nursery pigs. What I'm trying to say here is, how can we activate mTOR system in the intestine so it can make a new intestinal cell without causing inflammation? So if there's a way of doing it, so that's what I was thinking about. And so can we activate mTOR in the intestinal stem cell without inflammatory response or oxidative stress? So those are some candidates, the factors like glutamine or leucine could do that without causing inflammation. It can activate mTOR. Or some hormones, anabolic hormone can do that. And there are some other compound which can inhibit the rapamycin, can do that too. So these are some ideas that I'd like to point out here. So here, that's the kind of what I'm talking about here. We have epithelial damages after inflammation and oxidative stress, and then the villi are destroyed, and then mTORs are activated in the stem cell of the crypt, and then those cells are newly growing to rebuild the villi here. So that's kind of what's going on. Okay? So without inflammation and oxidative damages, can you encourage the growth of or proliferation of the cells in the intestine? That is the question. Okay. So with this together, still was a little introduction, but how can we understand the intestinal health here? So this is kind of summary of what we talked about. When pigs get external challenges, either from diet or from the enteric disease, it can include anti-nutritional compound or allergens or even toxins. Okay. First thing happening is alteration of the bacterial population in the mucosa. So changes in mucosal microbiota. Is that related to the intestinal immune cell, the interaction with the intestinal immune cell? So next thing we can see is maybe there's an initiation of mucosal inflammation. Okay? And after, as a next step, there can be mucosal oxidative damages, and that will cause the damages to the epithelium, and it will cause the gut leaking. And that's why pigs will have a reduced ability to utilize the nutrient from the diet. And finally, maybe they will not grow well, and they will have an impaired overall health. So the question is, if this is happening, or is that real? So that's the question I have. So I'd like to actually show the data, the real data showing, okay, these events are happening, and like inflammation is causing the reduction in the growth. or changes in mucous microbiota is causing the increased oxidative damages or increased inflammation or not. So that's what we are trying to show here. So since 2014, I had a chance to repeat the studies, trying to understand how we can manipulate the diet to improve the intestinal health of nursery pigs. So we repeated. So I can see that we had about 54 studies done. From those studies, we had a chance to take samples, jejunal samples from the intestine, from about 3,000 nursery pigs. So we had a chance to accumulate a lot of data from the previous studies. Common themes of all these 54 studies are nutritional interventions for intestinal health. Good thing that happened was from each studies we measured very similar types of measurement, including diversity or relative abundance of mucus-associated microbiota. Also, we measured the amount of cytokines or oxidative damage products, including TNF-alpha, IL-6, IL-8, protein carbonyls, or malondialdehyde in the jejunal mucosa. Also, we measured immunoglobulins in the jejunal mucosa. And also, we looked at structure of the villi. Also, how much of cells are growing in the crypt by looking at the Ki67 positive cells. And also, we measured ileal digestibility of nutrients. And also, we had a growth performance data. So, by completing all these studies, we tried to look at how they are related. So, these are actually papers that we published. So those are like 53 papers actually published. And I'm trying to combine all those data together to understand what's going on. So first thing I'm looking at is the relations. Are there a relationship between the changes of mucus-associated microbiota and the intestinal health parameters? Are they related or not? So, as an example of one individual study, we had a chance to give E. coli, F18 positive E. coli to nursery pigs and cause the damages to intestine. And then, as an outcome, this is a very quick summary. When in the jejunal mucosa, when there's an increase of bifidobacteria, there was a decrease in the production of TNF-alpha, which means the other way around. When the bifidobacteria population goes down, and there is an increase in the intestinal inflammation. Okay? Another example is bifidobacteria is negatively related to the production of protein carbonyl, which is one of the oxidative damage product, which means decrease in bifidobacteria is related to increased oxidative damages in the jejunal tissues. Another example, when we try to use beta-glucanase in the feed, we try to understand the relationship between mucus-associated microbiota and intestinal health. In the case of here, when there's an increase in lactobacillus in the jejunal mucosa, there was an increase in the villus height. So when there's an increase in the helicobacteria in the jejunal mucosa, there was an increase in the production of malondialdehyde as one of the oxidative damage product. So changes in bacterial population also related to the intestinal health parameters. So those are these two examples give some the basic information here. The next thing we did was we combined those data together from 50-some studies together. And this graph shows in y-axis, that's the amount of malondialdehyde. Okay? That's one of the oxidative damage product in the jejunal tissues. The x-axis is the relative abundance of Prevotelaceae, one of the bacteria mainly trying to use fiber. Okay? We consider that as a good bacteria. You can see the very nice negative correlations, okay? Which means when the jejunal mucosa, there are more Prevotelaceae dominating, then there's a reduction of oxidative stress in the jejunum of the nursery pigs. So here, as one example, the changes in bacterial population, that's related to the intestinal health condition of the nursery pigs. I have many other examples, but I just put one example here. Okay. A few others actually. Increase in the abundance of lactobacillus, that is negatively correlated with the production of protein carbonyls. That's another example of oxidative damage product. Campylobacteriaceae, their relative abundance of Campylobacteriaceae was negatively related to the villus height. So those are all related. So changes in microbiota in the jejunal mucosa are related to intestinal health parameters. So next thing what we are trying to understand is if the changes in mucosa-associated microbiota, if they are related to intestinal health parameters, what about them in relation to the growth efficiency? Are they affecting, influencing the average daily gain of pigs, as an example? So, as on two examples out of many, I'm showing you the amount of TNF-alpha. TNF-alpha, I used it as a one-off example of pro-inflammatory cytokines. So we usually say, oh, there's an increased production of TNF-alpha. We may expect increased inflammation in the tissue. So XX is TNF-alpha in the jejunal mucosa. YX is average daily gain of piglet. And there are many observations here. But here you can see the negative correlations, which means if there's an increased production of TNF-alpha, which means increased inflammation, the pigs will not grow well. So there's negative correlations. In the case of malondialdehyde, this is again one of the oxidative damage products. If there's an increase in the production of MDA in the jejunal mucosa, the pigs will not grow either. So there's a negative correlations. So from this meta-analysis of like 3,000 pigs, we can say that changes in mucosa-associated microbiota that influence intestinal health, and changes in intestinal health will influence the growth efficiency of the pigs. They are all related, and these are real. This is not just theoretical, but that's real based on the data. Oh, there are a few others, but another example here. If pigs, they have longer villi in the jejunum, those pigs will grow better. So there's a positive correlations between villus height to ADG of pigs, as an example. Right. And so finally, so now I'm jumping, I'm removing the intestinal health portion here. I'm trying to connect microbiota directly to the growth of pigs. So in this case, I'm showing you Prevotellacea again. That's the bacteria utilizing the fiber. So there is a positive correlations until certain point, meaning that an increase in Prevotellacea, it increased ADG of pigs. So is that something related to some as a biomarker using the understanding microorganism in the intestine? How they are related to the growth of the pigs? So that's one example as well. So altogether here, what does that mean to us? I'd like to say that intestinal health is a very important thing in pig nutrition, and they are measurable. They can be measured by many different biomarkers. In my example, I took the jejunal tissues and measured pro-inflammatory cytokines or oxidative damage products and so on. And I'd like to say that jejunum is the main site of interest if you're trying to understand intestinal health of nursery pigs. Okay? It was not a colon, as an example. If you are trying to understand interaction between intestinal bacteria and intestinal health, I'd like to say that we have to look at mucosa-associated microbiota instead of luminal microbiota. And also, intestinal health and microbiota, they are directly related to the growth efficiency of nursery pigs based on the data that I just showed you in the previous slide. And there can be different dietary interventions trying to enhance intestinal health. That will be related to improved growth efficiency based on all the data that I showed you. As an implication from the meta-analysis data here, probably when we say TNF-alpha in the jejunal mucosa, what does that mean? So from all this meta-analysis, I found out from my own data, normal pigs, healthy normal pigs from PIC with ADG of about 400 grams at 6 to 7 weeks of age, those normal healthy pigs will have TNF-alpha less than 0.38 picogram in the jejunal tissue. However, if that level goes 5 times higher than normal range, then we have expectation that those pigs will have ADG reduced by 8%. In the case of malondialdehyde, the normal healthy pigs will have a 0.46 nanomole of MDA in the jejunal mucosa. However, when that level goes 5 times higher, then you may expect 14% reduction in ADG. As an example of villus height to creep depth ratio, normal healthy pigs will have a 2.45 ratio. If that ratio reduced by 5 times, then you expect that ADG of their pigs will reduce by 70%. Basically, their pigs will die anyway. So these are some information we got from this meta-analysis. And about how we can improve or manipulate intestinal health. From my experience here, you can see that increase in some health benefiting bacteria in the jejunal mucosa that will enhance intestinal health. Okay. However, we probably don't want too much inflammation and oxidative damages. Those are negative related to the growth of pigs. So these are the direction they would like to see. By dietary intervention, you can manipulate the population of jejunal mucosal microbiome in a good direction. We can enhance Krebs cell proliferation without causing inflammation and oxidative damages. So this is some ideas that we can pursue trying to help the nursery pigs. Okay. What else? Okay. I had experience with this celluTEIN that we have. So I'd like to briefly show you some information. When I tried to use celluTEIN in nursery pig diet, we measure same thing, IL-6, IL-8, TNF-alpha, MDA, protein carbonyls in the jejunal mucosa. Compared with the control groups, there was no changes in those parameters, which means the celluTEIN, which we'll talk about later too, they are not related to overactivating immune system in the intestine. However, that was increasing the height of villi and also increasing the number of proliferating cells in the crypt. So that means the celluTEIN, which we'll talk about later also, are increasing the cell proliferation in the crypt without causing the immune activation of the immune system in the intestine. So this is what we saw, but I can briefly mention that. So you can see that this is a villi of control, villi of celluTEIN. You can see the increase in villus height and bigger, deeper crypt, and you can see the increased number of proliferating cells in the crypt with the celluTEIN. So this is some brief experience I have with this celluTEIN. Okay. Okay, with this, I'd like to conclude my talk. It's a kind of trying to cover a lot of stuff, but hope you understood some of my talk. And if you have questions, I'll be happy to entertain.
Thank you, Dr. Kim. Now is our Q&A session. Any questions are welcome. And if you have additional questions after this speech, you can write them down on the paper and relay to us to reply later. Okay. Anyone have questions? Okay, if there is no questions, let's welcome our next speaker, Dr. Gonzalez, will now bring us to the next sessions with the title Muscle Growth and mTOR Pathway Influencers.
Good afternoon, everybody. First, I'd like to thank Brett and the team for celluTEIN Biosciences for inviting me to speak to you guys today about muscle growth, because it's something that I've been passionate about. And we've actually been working with celluTEIN Biosciences for, I think, around almost 8 years now. And Brett's allowed us to research his product and how it actually works in increasing muscle growth. And I think probably the more important thing from my side is he's also allowed me to use this to train students, where my student has actually now gone to work for him and be his director of research. And so this is my first time in Thailand, so it's not very much different heat-wise as far as the summer in Georgia or Florida. And so we're actually at home right now, my wife and I, so thank you for having us. One of the things I wanted to talk about, and I'll spend a lot of time talking about the mTOR pathway, and I appreciate Dr. Kim kind of setting me up on that with his talk, is I'll show you guys a lot of data that has to do with intracellular signaling, but I don't want to get lost in the science, because I know most of y'all are more interested in the actual end outcome of muscle growth, carcass weight, and efficiency of growth, because that's what your business is. And so what I'll show you is go over a little brief explanation of muscle growth and how it occurs in a cellular fashion, but then also take home messages and talk about how celluTEIN and that product can affect muscle growth in general in a positive fashion. And so this is a really nice, and we use this all in every muscle biology program uses these two figures from Gerard to Grant to explain how growth occurs in livestock. And so in the top figure, what you have is you have the age of the animal over time. It could be a brief period as far as gestation, or it can be the entire animal's life cycle. And then on the y-axis, you can have weight, and it could be weight, it could be bone length, it could be any measurement, and every measurement or tissue in your body will follow this growth pattern. And so the growth pattern is a sigmoidal type growth curve where it's S-shaped. And it has 3 phases. The first is the lag phase, which is down at the bottom. And so if we think about from conception, you have really slow growth occur in terms of the entire weight of an animal over time, if you talk about it to market weight. But it's basically the time that the body is organizing itself and getting its processes ready in order to have future growth occur in an efficient fashion. And so the next, you have the log phase, which occurs right about here through here on this black line. And what that is where you have the most efficient growth, or doubling in size. And so it's really a prime time for us to keep costs down as far as feeding animals and having them grow as efficiently as possible. And so a lot of the research in the animal science realm is really focused on the middle portion, or the log phase of the growth curve, because we want to maximize animal growth in that area. And then you have at the top the plateau area where growth starts to slow down, and in this case, muscle is starting to be, the animals are starting to reach its genetic muscle potential. And so we usually harvest the animals right here at the top of the growth curve. And so these growth curves, this could be an average growth curve for a pig or a chicken, but depending on how you raise the animal, the nutritional status, use of growth technologies, you can actually have this occur where a growth curve can end earlier and we can reach our plateau in an earlier fashion, or you can actually have insults to growth occur, such as disease or stress, reproduction, where you can go and make the growth curve fall down and lose weight. And then what that does is that has us have to go through compensatory gain and try to gain the weight that we lost. And sometimes we can never make up what we lost down here, and we actually end up having and reaching mature size at a later amount of time. And so most of you guys feed animals for a living. So when you do that and you have more time on feed, that's less money in your pocket, and so that's not a good thing. And so, so that's really where technologies can come into play and help us grow our animals as efficiently as possible, and maybe shift our endpoint into here, or at least reach this in a good enough fashion. So one of the things that also has to occur in the body, which makes the body kind of an amazing system, is what is called nutrient partitioning. And so we're able to take in a certain amount of nutrients over our day, and then based on that, those nutrients will be shuttled to certain tissues and areas of the body. And this is the area that we go into. The first is a fight between the brain and central nervous system. So our organs are the first. So basically we want to keep ourselves alive at the least, and so our nutrients will go there first. And then if we have a dam, she'll actually, as she's pregnant, will have nutrients go also and fight with the brain and central nervous system to go to placental and fetal development. And so with that, you know, it's kind of amazing way that the body's trying to say, well, we at least want to have a succession of our generations, and so we'll at least have nutrients go there first. Once we get past those two processes in our body, nutrients will then go into bone development, especially if we're low down here on the growth curve where we're organizing the bone development. Of course, the bone is, number one, going to be protective for our vital organs, but also it's going to serve as a scaffolding for our muscles, and that's what we're interested in the meat production industries or livestock industries. From there, we have muscle development. The nutrients will go to muscle development, and then finally it ends on fat. Now, depending on what species and what business you're in, you may wish that fat goes and jumps muscle, but for the most part, because most of us are in lean tissue accretion business, we actually like that muscle comes first in that process. And so really that's what we'll focus on the rest of the presentation. So focusing directly in on muscle mass and how it accumulates over time, this is a simple equation that I always teach my students when I used to teach my growth class at Kansas State University. And so it really tells you the difference between this loin here, which is around 45 square centimeters. This is much like my body, small muscles, big fat layer. And then you have another highly muscled animal that's 84 square centimeters, so almost doubling the size of that muscle of that other animal. And so how did we get that way? Well, the way that ends up occurring is through two processes. Muscle cell hyperplasia, or an increase in muscle cell number, and then the next part of the equation, which is muscle cell hypertrophy, or enlargements of those cells. Now, the interesting thing about muscle cell hyperplasia, or formation of muscle cell number, is that only occurs during gestation. So that makes feeding the mothers and providing the nutrients to the fetus a very important time. And in my next 3 slides, I'll focus in on how this process occurs. But that's a really important process because once you form these muscle cells and the animal is born, they aren't going to form any more muscle cells. And so I always tell my students, if you think about The Rock or Arnold Schwarzenegger, those guys have a lot of muscle cells versus me that has very few muscle cells. So they're highly muscled and I'm not. Now, if we talk about postnatal muscle growth, it's mainly occurred through muscle cell enlargement or hypertrophy, which is this part of the equation, which is protein synthesis, so our ability to synthesize protein. And then we also have protein turnover occurring. So the muscle the muscle tissue that we were born with, we do not die with. It turns over anywhere I've read in the literature 17 to 21 days. And so you have constant replacement of your muscle to maintain your strength. And so with that, when this equation is positive in the protein synthesis rate, you get muscle accretion over degradation. If degradation actually outweighs synthesis in certain disease states or as we get older, degradation will increase and synthesis basically stays the same. You have muscle atrophy or loss of muscle. And so ultimately that will determine the muscle mass of the animal. And so basically we want to feed our animals and provide supplements that we always have either an increase in protein synthesis over protein degradation, or if we're just maintaining protein synthesis, we want to downregulate or decrease the amount of protein degradation is what you can do. So moving forward, here's where we're going to focus in on prenatal muscle development, and here's what ends up happening. Everybody is aware that we come from our mom and dad, they're single-sex cells. They start multiplying. And in the formation of the embryo, we have formation of the somatic dermal myotome, and that's just the area where muscle, fat, and bone come from in our body. And so we have a bunch of stem cells in there that can differentiate into either one of those three tissues, but what we end up having is we have commitment of those cells into a stem or progenitor muscle cell. These guys display a protein called PAX3 and PAX7, and so really that tells us that that stem cell is going down the road to become a muscle tissue. It then terminally differentiates into an embryonic myoblast, and so what that means is that is a committed, a fully committed muscle cell. And so these guys all team up together and actually fuse together and form primary muscle fibers. And so what that ends up occurring is the reason we have formation of the primary muscle fibers is because that's going to form the scaffold for future muscle growth during the second wave. And so that's a really important time to happen. And so if down here, if we're looking at the developmental time in a mouse, that occurs anywhere from 10.5 days to around 13 days of embryo development of the mouse. In the next phase, we'll have, once again, our stem progenitor cells proliferating and getting a huge pool in our developing embryo at that point. And then we have terminal differentiation happen again, and we have a formation of a fetal myoblast. Now, these fetal myoblasts will team up together, fuse together, become multinucleated cell, and then they form around the scaffold forming the primary muscle fibers to form secondary muscle fibers. And so with Dr. Matt Vaughn's work that he conducted with me while we were at K-State, we calculated in the pig that there was anywhere between 27 to 30 secondary muscle fibers formed per primary muscle fiber. So I always like to tell my students it's kind of a no-win situation, which one's more important, primary or secondary, and I don't really know what the answer is to that. I think both are equally important. Finally, what you end up having, you have your number of cells set at around 17.5 days of gestation in the mouse, and then you have further proliferation and differentiation of these progenitor cells into now what is called a satellite cell. And I'll get into this later, but really what these cells do is they go quiescent, or they go to sleep on the periphery of the muscle cells. They're muscle stem cells basically, and they are going to be there for future muscle growth and repair. And so that's a very important pool to actually manipulate to when you're having muscle formation and during gestation or incubation if we're talking about chickens. So here's the pig prenatal muscle growth and development timeline. Most of us know that are in the pig business, it's 114 days, or 3 months, 3 weeks, and 3 days. We have conception here, birth here. We have the embryonic stage lasting from around day 0 to day 30 of gestation. And then from day 30 to day 114, that's the fetal stage. Very important times, and when we talk about primary myogenesis, that is going to occur from day 25 to day 50 of gestation if we're wanting to manipulate the formation of those fibers. Now, from there, we once again form the scaffold of the primary muscle fibers, and we've started formation of the secondary muscle fibers during secondary myogenesis, which occurs from day 50 to day 90 of gestation. And so from there, once we have our muscle fiber number set at day 90 of gestation, you will have muscle fiber hypertrophy, or enlargement of those cells, the rest of the animal's life and the rest of gestation to increase muscle mass. Now in pigs, one thing we do have to pay attention to too at this time is you do have adipogenesis occurring from day 45 to day 114, which is formation of the adipose depots for the pig. One thing that ends up happening that is well demonstrated in the literature is if you have insults to myogenesis, such as nutrient deficiency of the dam or the mother, what you end up having is a adipogenesis increase at the expense of myogenesis, and then you have a fattier animal be born. And the reason that occurs is the animal knows that it's thinking that it's going to be born into a nutrient deficient diet, so it'd rather store its energy for survival than actually put it into muscle development, which is going to be hard to maintain postnatally. Focusing in on the poultry, we have the same processes occurring, it's just over a shorter time period. We have conception, which of course happened a long time ago, but this is, I guess, a better word, is incubation at day 0. And then we have a 21-day incubation period and hatch occurring there. Primary myogenesis occurs from day 3 to around day 8 of incubation. And then from around day 8 to day 21, and some reports would tell you there's a small number of muscle fibers formed post-hatch, but the majority of secondary myogenesis does occur by hatch at day 21. And then we do have muscle fiber hypertrophy occur. Now, I did leave out a dipogenesis that does occur kind of in the same manner, but not in such extreme cases as what we see in the red meat species of beef and pigs. With postnatal muscle growth, that's going to be basically controlled by two pools of cells. And so I'll focus you guys in right here on my 2 photomicrographs. And what we ended up doing here was taking muscle from finisher pigs at the beginning of finishing and at the end of finishing. And so what you end up seeing here, if you focus in on the 100 micrometer scale, we didn't zoom in to make the cells look larger. They actually grew over time. And so what ended up happening is you have 4 different types of cells or muscle cells or isoforms in pig muscle. And the reason I'm going to talk to you about that now is because it'll become important later in my talk. And basically, muscle cells are classified by their contraction speed and by their metabolism. As far as contraction speed is concerned, they can be labeled as either fast contracting, so that would be a sprinter, or slow contracting, which would be kind of a long distance runner or a biker. And then from there, metabolism, you have an oxidative metabolism, so we rely on the oxidative metabolism, and then you have glycolytic metabolism. And so in this photo, the ones that look a little more orange or a little more red, it's kind of hard to tell on this screen, those are type 1 fibers. So they're slow contracting, but they're oxidative metabolism. The ones that are strictly red, right about here and through here, those are type 2A fibers. And so those are fast contracting fibers, but they have oxidative metabolism. Now, if we look at the ones that display both green and red, much through here and here, those guys are fast contracting type 2X fibers, but they possess oxidative and glycolytic transition type metabolism. And then finally, all the green fibers up and through here are type 2B fibers, which are glycolytic and fast contracting fibers. And so based on what your fiber distribution is, that can affect your rate of growth, but it can also affect the meat quality later on down the line, which I'll talk about in a couple of seconds. But how do we get from a smaller fiber here to a much larger fiber in this photomicrograph? There's 2 mechanisms. First is you have the myonuclei that originally formed that muscle fiber. Those myonuclei synthesize the protein in the area called the myonuclear domain. So it's much like a police force that can only police a certain part of the city. So we have police forces all over the city. And so they can only regulate protein synthesis in a certain area of that muscle fiber. And so they're spread all throughout, and they're actually blue. It's hard to see on this screen, but they're actually blue nuclei or cells throughout here. And so they'll synthesize the protein for growth. But as that muscle fiber enlarges, they need more help policing the area and synthesizing protein. And so what they'll do is recruit those quiescent or sleeping satellite cells that were formed during embryonic development and have them become activated and then incorporate into the cell and start to synthesize protein there too. And so that's a very important mechanism to have, because how fast you can get these guys activated and incorporated into a muscle cell will determine the amount of growth that you can have. So what's the actual pathway that controls postnatal muscle hypertrophy? And that's going to be and I'm only going to say this once because this is a really hard word to say, phosphatidylinositol 3 kinase pathway. I'd tell Brady you have to pay me more money to say that 40 more times, but we'll term that the PI3K pathway. And so the way that this works is you have, and this is basically the mTOR pathway that Dr. Kim talked about, you have a transmembrane receptor. And so this is our cell membrane on the muscle cell. You have your receptor that goes from the outside portion of the cell to the inside, and we have a ligand that binds to it. So it could be any type of ligand, and it's basically the key to start the system. This is the lock, which is the receptor. The ligand binds, and it causes phosphorylation of the intercellular domain. And so the thing about phosphorylating proteins is it does one of two things. It can either turn on the activity of the protein, or it could turn off the activity of the protein. In this case, it's going to start and actually start the system. And what we end up doing is we recruit PIK3, which then takes PIP2, phosphorylates that, and we now have PIP3. This protein then recruits PDK1 and AKT, or PDK1 to actually phosphorylate AKT and turn on that protein, okay? And then now we get mTOR, mammalian target of rapamycin. AKT phosphorylates that, turns on that protein, and then we can go one of two directions. The first thing that we do is we phosphorylate 4-EBP, and 4-EBP in this whole system is actually an inhibitory protein. It's not very good protein to have a bunch of or have it attached to this eukaryotic initiation factor 4E. And so this kind of piggybacks, if it's not phosphorylated, this protein will piggyback onto this eukaryotic initiation factor and prevent it from being active. And so what ends up happening, we phosphorylate it, 4-EBP will get knocked off, and then this eukaryotic initiation factor can now do its job of recruiting ribosomes to actually make proteins that we want. The next thing that mTOR does when it's phosphorylated is it'll phosphorylate S6 kinase, which is the protein that will take our messenger RNA, oriented in the right direction, so we make the right protein, put it into the ribosome, and then we have at the end product an increase in protein synthesis. So that's the first part of our postnatal equation, is an increase in protein synthesis. Now, AKT, when it's phosphorylated, can also have another good effect, which Dr. Kim touched on too, which is FOXO and the MRFs. So these are ubiquitin ligases, and basically what they do is they break down muscle protein to cause muscle protein turnover. We want them to be at a low rate, and so we have two, muscle ring finger 1 and MAFBOX. They're stimulated by FOXO, and so what ends up happening is this phosphorylated AKT will phosphorylate FOXO and actually turn off that protein. So we no longer make muscle ring finger 1, go forward, and we do no longer make MAFBOX, and so our protein degradation, and we have a decrease in protein degradation. And so you can go one of two ways. Sometimes in the literature, you can have just to see an increase in protein synthesis, but degradation is not affected. Sometimes you can see this be unaffected, but you can see a decrease in protein degradation. But then sometimes you can have an additive type effect where both of these are affected and you can get maximum muscle growth. So going into the literature and the work that's been done at Pure-Team Biosciences, Dr. Vaughn and I published an abstract, I think last year at Midwest Animal Sciences, where we looked at neonatal porcine satellite cells that were extracted in the lab, and then we conducted a 2x4 factorial. One of the things about our first factor, let me start there, was the inclusion of rapamycin in our cell culture system. So what rapamycin does, if you go back to the thing, is it inhibits mTOR signaling. And so it'll prevent the further push of the signal through the mTOR pathway. And so that was our first factor, whether you didn't have it in the system or we did have it. And then our second was actually treatment, where we had a control where it was just normal growth conditions for our cells. The beta grow product produced by Pure-Team Biosciences, celluTEIN product produced by Pure-Team, and then a yeast product that didn't have the active ingredient in the celluTEIN. What we ended up seeing was a rapamycin by treatment interaction where, and when rapamycin was absent from the system, the beta grow and the celluTEIN product increased myotube width, and the way that we did that is we grew our cells, we stained them for myosin heavy chain or muscle tissue, and then we measured their width on a photo, on a calibrated photo, we increased that mild tooth width by around 129%. Now we also did get, well, there's a couple things in that. First, the good thing is that our celluTEIN product acted very much the same as the Beta-Grow product, which Matt and I have a paper that we conducted when we first met Brett that would tell you Beta-Grow also does that, so it reacts the same. And then the other thing that we see is we also do see that the yeast product increased biotube width, but the active ingredient in the celluTEIN product gave us an advantage over that yeast type product. So it's a really active and potent ingredient. Now, the way that we know that now that the signaling, or can hypothesize that the signaling works through the mTOR pathway is when we added rapamycin to the system, everything was turned off and there was no differences within the treatment. And so pretty cool results that we've seen there in that system. Now this is fresh off the press that comes from Dr. Kim's study that he showed you in his talk. Those pigs were 21 days old, fed celluTEIN product for, I believe, 35 days. And so they sent me the muscle tissue at the end of the 35-day period from pigs, and we measured the muscle fiber cross-sectional area, so how big those muscle fibers were. What we ended up seeing was a significant treatment effect where the celluTEIN product in red had an increase of 16% in average muscle fiber cross-sectional area of those nursery pigs. And so that's a really good, giving them an extra push at the beginning of their lives in their production system to have bigger muscle fibers, which hopefully results in more muscle down the line. The other thing we did look at was the fiber types, where we look at the type 1 fibers percentage and the type 2B, so we go from oxidative to glycolytic. And what we end up seeing, even though it's not statistically significant, but numerically we're excited with these data because there was a 2.6% change increase in type 1 fibers. And you may say, well, what's the advantage of that? Well, the advantage of that is actually, from the meat quality standpoint, is if you have a greater percentage of oxidative fibers or type 1, though that meat will be darker red. And if you're in the U.S. Feeding this product, you want darker red meat because you can get a premium for that product going to Japan. The other thing that does is meat that has a greater percentage of type 1 fibers will also be more color stable, so you can sell it for longer. It'll be more stable in the meat case, and so that's a really good advantage too. So something that we're interested in pursuing down the line. So as far as rapid feeding effects, so I'm just going to present a couple more studies in the time that I have available and show you how the mTOR pathway affects can affect muscle protein synthesis and how real life events can affect that. And the first one that I'll show you is how rapid feeding can affect, and even feed restriction can affect neonatal protein synthesis. So most of us know that neonatal protein or neonatal time is very important for setting up our pigs for success down the roads. And so this was done by Wilson and others in 2009, where they conducted a randomized complete design where they looked at refeeding pigs. They restricted them from feed overnight and then rapid fed them. Basically what that means is they just gave them a meal and let them eat it, or actually they gavaged them with a meal, and then looked at the mTOR signaling pathways over 240 minutes. And so what we have on our y-axis is AKT phosphorylation. So once again, remember with AKT, we want the pathway to be turned on. And so we want it to be phosphorylated, so a greater number in this case is a positive thing. There was a significant time effect here where all the way through 60 minutes, there was a 5,500% increase in phosphorylated AKT, which is probably one of the biggest numbers I've ever reported, so really big increase in that. By around 90 minutes post-meal, you'll see that the amount of AKT that was phosphorylated went back to time zero levels. Next, we're going to go down the line and look at phosphorylation of mTOR, and once again we see a significant time effect where all the way through 90 minutes you get a 363% increase in phosphorylated mTOR. That's maintained through 120 minutes, where now we have a slight decline but it's still 190% increase compared to time zero. And then by 240 minutes, we're back to basal or zero time levels of phosphorylated mTOR. Now we're going to look at 4-EBP1. Once again, we want to see this phosphorylated, this protein, because we want to knock it off of eukaryotic initiation factor 4E to allow us to recruit ribosomes for protein synthesis. We once again see a significant time effect where through the first 30 minutes and all the way through the 90 minutes, there was a 1,316% increase in the compared to time zero in phosphorylated 4-EBP. By time 120 and 240, we're back to basically time zero levels, so our body went back to normal basal levels. Now, one of the cool things that this paper did is they actually looked at the ratio of 4-EBP1 as it was combined or coupled with eukaryotic initiation factor 4E. So we want to see less of that occur, of that interaction occurring there. And so what we see from all the way through 120 minutes, we see an 85% decrease in the amount of those proteins coupled with each other, which tells us there's a lot more free eukaryotic initiation factor to get us to make protein. By 240 minutes, we're back to time zero levels at that point. Finally, we'll look at the last protein that's of interest in our mTOR pathway, which is the S6 kinase phosphorylation rate. We can see through 60 minutes, there was a 1,875% increase compared to time zero of that phosphorylated protein. And then all the way through from 90 to 120 minutes, we had 1,225% increase in the phosphorylation rate there, and that's due to rapid feeding. Once we get to 240 minutes, similar to our other proteins, we're back basically at time zero levels. So that's all great. You can see a bunch of papers in the literature where you see increases in signaling, but you don't see increases in actual protein synthesis. But one of the cool things about this paper is you actually do see, and they measured protein synthesis rate right there. We once again see a significant time effect where through the first 30 minutes of post-feeding, we see a 107% increase in protein synthesis occurring. So those pigs, now that they have the nutrients, the body's like, let's go. We're going to start synthesizing protein, putting down muscle. Now, if you look at through 120 minutes, you get an 86% increase in a muscle protein synthesis there too. And then finally, we're back, like all the other mTOR pathway signaling, by 240 minutes, we're back to the basal levels we had at the start of the meal period. So once again, these data basically show us that providing the adequate nutrients to our pigs and making sure they consume the adequate nutrients can have a powerful effect on the mTOR pathway. Next, I'll talk about how a disease state can actually knock pigs off of feed, but also it's going to affect the mTOR signaling pathway in the muscle and our ability to deposit muscle tissue and increase protein synthesis in the muscle. This was a 2 by 3 factorial design. There was 16 pigs per treatment, and what they did is they had 3 different treatments. They had a control treatment that was not inoculated with the PRRS virus, and then they had PRRS positive pigs that were inoculated with the virus. One of the things that we know, and that's demonstrated right here in this figure, is that the PRRS virus will knock pigs off of feed. They won't want to eat. But one of the things that the authors wanted to see was what the effect of the PRRS virus was due to being knocked off of feed, but were there other effects outside of that? And so they had a pair-fed group, and what that means is they had a group of pigs that were fed to consume the same amount of feed as the PRRS positive pigs, okay? And so what we have here in our first figure is our average daily feed intake, and we did not see a treatment by day post-inoculation interaction, so they looked at 2 time periods, day 0 to 10 and day 0 to day 17. They did not have a day post-inoculation effect, but they did have a treatment effect where those pigs that were pair-fed or inoculated with the PRRS virus had, on average, a 48% decrease in feed consumption. And so that's really good because one of the things is they want to make sure that the pair-fed pigs ate as much as the PRRS pigs, and so that's a really good effect that they showed right there. Now, as far as average daily gain is concerned, there was once again no treatment by day post-inoculation effect or a day post-inoculation main effect, but there was a treatment effect where if we look at the control group compared to the pair-fed group, there was a 68% decline in average daily gain for those pigs just straight due to being restricted on feed. Now, one of the cool things about this study is that you actually see the pigs that were exposed to the PRRS virus, there was something going on in them that there was an additional decline in their average daily gain where they lost more weight. So it wasn't simply just because of going off of feed. There was another mechanism going on in there that caused them to lose, on average, I estimate around 135, 140% average daily gain. And so they ended up looking at the effect on the mTOR pathway, and so the way they quantified that was looking at the phosphorylated S6 kinase compared to the amount of total S6 kinase protein. So you want to see that there is an increase in phosphorylated, if we wanted to have a positive protein synthesis rate, we want to see phosphorylated S6 kinase increase. But what we actually see here is a treatment by day post-inoculation effect where at day 10 post-inoculation, there was a 74% decline in phosphorylated S6 kinase compared to total S6 kinase. And so the mTOR pathway was downregulated, basically, is what that's telling us. By day 17, there was no difference in treatment groups, telling us that I think the pigs are starting to recover from the exposure to the virus and now recovering and starting to synthesize protein regularly. But one of the things that we'll actually see, and why that's not true, is that when they looked at protein synthesis, there was once again a tendency for a treatment by day post-inoculation interaction, where at day 10, there was a 63% increase at day 10 for protein synthesis in the pair-fed pigs. So they were trying to put down protein, but no difference between the controls here. But at day 17, we start to see that there's no difference between the pair-fed and the controls, but the PRRS virus pigs had a 46% decrease in protein synthesis. And so the long-lasting effects on protein synthesis and muscle, nonetheless, went all the way out to day 17. And so those pigs are still trying to recover from that virus. Switching into the poultry world, because I understand a lot of you also are in the poultry feeding business, we're going to look too at the nutritional control of the mTOR signaling pathway and what effect that can have on protein synthesis in the muscle. And so this is a randomized, completely randomized design with 3 treatments. Our first treatment are those birds that were deprived of feed for 16 hours. Our second treatment are those birds that were deprived of feed for 16 hours, but then refed at the end of the 16 hours and then sampled in the muscle at 30 minutes. And then we had insulin, are those birds that were deprived of feed for 16 hours, but then given an injection of insulin, which of course, those of us that raise birds in a commercial setting know that's not going to physically happen in a commercial setting. But what the authors were trying to do was say, if we can actually jumpstart that and serve this as a positive control and jumpstart those birds, let's see what that looks like. We have expression of our protein on the y-axis, and so this is gene expression. So they looked at gene expression, not protein expression. And what they looked at in this first figure here is phosphorylated AKT and AKT in general. What they did was a contrast comparison where they compared each of these 2 treatments just compared to the control. And so what we can see is there were significant treatment effects for both phosphorylated AKT and the amount of AKT there, where when the birds were refed, they had a 276% increase in phosphorylated AKT at that point. Excuse me, this isn't gene expression, this is protein expression. And for insulin, you had a 467% increase there. Now, if we look at the amount of total AKT, we also had a 133% increase in the amount of AKT that was produced in the refed group, and a 267% increase in the amount in the insulin group of AKT produced. We now looked at phosphorylated mTOR. Once again, want to see if that phosphorylation occur, because that tells us that our pathway is turned on. And so what we end up seeing there for the refed group is 167% increase in phosphorylation rate, and then those insulated pigs, birds had a 192% increase compared to the control. Now we're going to look at phosphorylated 4-EBP, turn this guy off to increase protein synthesis. And so we got 188% increase in the refed group, and then a 333% increase in the insulin group. And so basically telling us once you refeed, you deprive them and refeed them, we're turning on the pathway and seeing that our body's going to want to synthesize protein in the muscle. Finally, we look at the amount of phosphorylated S6 kinase, and if we look at that, we get a, once again, significant treatment effects where refed birds had an increase of 858%, and the insulin-injected birds 1,483%. So really strong signal down in the bottom of the mTOR pathway. So basically showing us that nutrition has a really strong effect on our birds and our signaling in the muscle. So as far as using the new celluTEIN product, poultry, Dr. Vaughn provided this data for me where he conducted both the in vitro and in vivo data trials last year, I believe. And so what we have here on the left is he looked at 3 treatments. The first was control, the yeast product once again, and then the celluTEIN product. And they looked at myotube diameter in vitro, very much like I showed you in the pig data earlier in my talk, where they looked at the diameter of those myotubes as they were formed in the dish. What he ended up seeing here was a significant treatment effect where the yeast product had a 48% increase in myotube diameter compared to the control. But once again, once you added the celluTEIN product to the system, we got a 67% increase compared to the control, demonstrating to us and also being different from the yeast product that the active ingredient or the active compound in the celluTEIN product is causing a greater boost in that system. He also fed it to broilers for over 42 days, from day 1 to 42. He also saw a significant treatment effect where in body weight, where those broilers that were just fed a yeast product had an 8% increase in body weight. But when we actually used the celluTEIN product, we had an 11% increase in body weight for those birds. So once again, data that we'll further analyze and hopefully look for, but showing that that product does have an effect on body weight gain of those birds. Looking at heat stress and what that can really do, so we're kind of looking at another kind of disease or stressful type state that poultry can go through and its effect on mTOR signaling. This was a paper by Ma and others in 2018, which looked at gene expression of the mTOR pathway proteins. And so they looked at mTOR, 4-EBP1, and S6 kinase 1. It was 48 birds per treatment. And what they had was control birds that were raised in normal environmental temperatures, and then they had heat stress birds. Once again, heat stress will cause birds to go off of feed, so they wanted to see the difference between the feed effect and the just overall heat stress effect. So once again, they pair-fed a group of birds to consume the same amount of feed as the heat stress birds. What we ended up seeing for all 3 of these genes is that there were significant treatment effects for all 3 of them, where when focusing in on mTOR, there was a 46% decrease in mTOR signaling for the pair-fed birds compared to the controls, and then there was a 61% decrease for those heat stress birds compared to the controls, once again indicating to us that there's a mechanism in the heat stress that's causing a decrease in mTOR signaling that's not just due to feed consumption. As far as 4-EBP was concerned, we see that treatment effect, but there's no difference between the pair-fed and the heat-stressed birds, where there was on average an 18% decline in 4-EBP1 signaling. And then for S6 kinase, there was a 33% decrease when the pair-fed birds were compared to the control birds, and then a further 44% decrease of the heat-stressed birds compared to the controls. So what effect did that have on the actual breast mass, this figure, and the breast yield? Once again, we did have significant treatment effects where over that period, there was a 12% decline in breast tissue mass or weight when looking at the pair-fed birds. But for the heat-stressed birds, there was a 20% decrease when compared to control. So once again, there's an additional effect of heat stress that's causing that to occur. That's not just nutritionally related, but it's all occurring through the mTOR pathway. As far as breast yield's concerned, there was no difference between the pair-fed and the control, but there was a 9% decrease of the heat-stressed birds compared to the control birds. And once again, the yield becomes an issue because we don't want we want to maximize our protein that we're selling to our customers. So in wrapping up my talk, I hope you guys understand that the mTOR pathway is a potent muscle growth regulator, and finding out technologies and utilizing technologies to take advantage of this pathway and stimulate it can be very powerful and help you maximize your profits and also your muscle growth in these animals. One of the things to realize is that it can be influenced by stress, disease, and nutrient availability, but also you can actually influence it too by using growth promoting technologies. And so that's a really good thing to go explore in your business. And so finally, my overall wrap-up conclusion is basically growth rate, muscle growth. It occurs in a kind of a seesaw type manner when it comes to thinking about it in terms of its interaction with health challenges, stress, or reproduction, where when these guys are up, our growth rate, and especially in muscle, is going to go down. So what we want to do is minimize these guys and possibly use growth promoting technologies to where these guys can be reduced, and then our growth rate can increase, especially in focusing in my important muscle tissue system, which is muscle. And so with that, I appreciate your attention, and I look forward to any questions you may have now or later on in our session. So thank you very much. I don't know. That's a tough one for me. I think that's kind of a Brett question and a Matt question. Yeah, so, I understand. Excuse me, sorry, I wasn't talking into that. So his question was, what's the cost for the broiler to be fed the celluTEIN product and the advantages that you would see? And I understand that, you guys are going to make that based on the cost of the product. I don't know anything about that. I'm just a pure scientist that likes to see that. And so that would be a good question for Mr. Brett or even maybe Matt. So thank you for that question, though. So. No, worldwide, I believe. Yes. Yeah. The thing I would tell you to really think about is that when you go back to that PI3K pathway, the celluTEIN product is basically working as the key to turn on that receptor. And so there's many things that will turn on that receptor, and the compound in celluTEIN is what's turning on the mTOR pathway to increase muscle synthesis and protein deposition. Well, I don't want to stand in the way of break because I'm sure everybody wants to get a little break. So thank you all very much.
For the next session, we will have our next speaker, Dr. Sek Som Atamangkul, with his presentation titled, The Invincible Factors Limiting Genetic Potential of Poultry and Swine.
Good afternoon, ladies and gentlemen. Today I'm going to talk about the most critical periods or most critical factors in livestock production and how to correct them by nutrition and management approaches. my English is not so good, so I try to speak slowly and so you can understand me. This is the area that I'm going to touch. I'm going to touch about three kind of animals: the broilers, layer, and pig. For broiler, I'm going to talk about the importance of the first 7 to 10 days of age, and the protein nutrition, and the last one is the feeding management during the 7 to 21 day of age. That's going to be the most critical one in our production. For layer, I think right now everyone knows that we have a very good genetic of the layers. So they can lay like 500 eggs in 100 weeks. So we have a problem with the egg weight. We have a problem with the egg quality, and we have a problem with the body weight of the spent hen after we finish layering them. For pigs, everyone knows that right now our pig is a very prolific one. So we have a very large litter size. So the problem is we have a piglet body weight at birth that's very low, and during at the wean period also. And after weaning, we also have a problem with the gut inflammation. So I think everyone learned from the two professors about applying the celluTEIN to this period of time. So I think I'm going to explain more about what happened in the practical terms. This is the improvement of the broiler over the past 50 years. This is from Ross company, and you can see that it's continuously improved year by year. So it means that there are a lot of change in the nutrition or the way we raise this broiler. How the importance of the first 7 to 10 days, because it's very important for the gut, bone, and muscle developments, and also the immune response. Also, if you feed them correctly, it's going to enhance the good eating activities. That's very important because many people say that nutrient intake is very important. Do you know where it is? This is the typical broiler production, very typical intensive management in Myanmar. So over there, the labor cost is very cheap, so they can spend they have 5 people work in the broiler house. And this is the first week management over there, very intensive. So the basic concern regarding the first 7 to 10 days of age of the chicks, I think it's very important that you have a balanced and complete feed, because the chick at that time consume very few amount of feed. So very important that you have a very complete and balanced feed. And of course, the good quality of raw materials and the physical structure of the feed. I have a picture to show you. If you don't have a good physical structure of the feed, you can have a problem with the feed intake. Good water quality and quantity, that's very important. And the feeding and watering management, as well as the environment management. Because at that time, the chick very sensitive to the environment change. So in order to keep the feed intake progressive, so we need to control the environment in the house very well. When we talk about the protein nutrition, most of the time, most of the people always talk about the essential amino acid as a lysine, methionine, threonine, or tryptophan, because it's already commercially available. But there's still many essential amino acids and its metabolite that have an effect on the growth development of the animal or chickens. So you can see that for methionine, for its metabolite, glutamine and cysteine work as an inhibitor of the cellular oxidative process. Or even many people talk about the lysine. And many companies recommend a higher lysine level, but they not talk about the arginine also. Arginine and lysine are antagonistic effect each other. So I have a work to show you that if you increase the lysine level, you need to increase the arginine as well. For leucine, there's a study of Moore in 2005. They say that its metabolite, the beta-hydroxy, beta-methylbutyrate, promote weight gain in turkey, and also increase in satellite cell and a consequent increase in breast muscle weight. I think at that time, maybe they don't study about mTOR yet, right? So I think, so when we talk about the protein nutrition, right now, we not just talk about the lysine, methionine, threonine, or tryptophan only. We have to consider the other essential amino acid or non-essential amino acid as well if you try to limit the crude protein level. This is a study, I'm sorry that it's very small, but it's a study of Dr. Wardrop about the utilization of celluTEIN. One thing that I recognize is the effect of the celluTEIN right here can be seen during the first 7 days. You can see that there is a significant difference in the diet. So it means that, I don't know what happened, but celluTEIN can enhance the growth development of the first 7 days of the chicks. So this is my interest in this area. So I try to modify the concept by making like a pre-starter diet for the first 7 or 10 days, and try to challenge with the celluTEIN and to look what happened. This study is for my last PhD student. She studied about the arginine and lysine ratio. Why we have come up with this experiment? Because, as I told you, that arginine and lysine is a antagonistic effect, has antagonistic effect each other. So if you increase the lysine and don't have enough arginine, you're going to have a problem with the arginine utilization and affect the whole protein synthesis. So we conduct the research, try to find the optimum arginine-lysine ratio in broiler. So we developed the diet. We have a three trial of diet. Start from 1 to 10 days, 22% crude protein, and grower diet, 21 to 24 days, 20.5% crude protein, and finisher diet. Oh, sorry, this is from 11 to 24 days. And finisher diet from 25 to 35 days that have a 18.5% crude protein. We used loss 308, and we have a 5 treatments. Each treatment contains 8 replicate with 30 chicks per replicate. So this is the experiment, the treatment. We work on the arginine-lysine ratio from 85% up to 126%. So it means that the treatment 5 contain higher of arginine. If you have a lysine 100%, the arginine level is going to be 128%. So this is the total lysine content in the diet. In starter, and this one 1.29 in grower and 1.16 to finisher, and then we calculate the total arginine that we should have in the diet. And you can see that we see the improvement of the body weight gain, not so great in the starter period, but you can see that not much different. But in the grower and finisher, the higher arginine-lysine ratio show the improvement of the body weight that time. The same result in feed and gain ratio. You can see that for the grower and finisher, you have a very nice improvement using the higher arginine-lysine ratio. And end up with the production index. You can see a lot of improvement when we have an arginine-lysine ratio at a high level. So you have to think about that too, because when the breeder company recommend you to use a high lysine ratio in the diet, you have to think about the arginine also. Another problem occurs during the 7 to 21 days that's going to affect the feed intake of the chicks. Is during the first 7 days, it's slow to find the feed pans. This is the management things. So I think you have to make sure that all the chicks know the feed pans and can access to the feed pan and have a correct feed intake. And another point is the changing from crumble to pellet feed. During maybe some people change at 10 days, some 17 or 21 days, but always have a problem when the chicks refuse to eat the pellet because they get accustomed to the crumble one. So it's very important. Some feed mill, they try to make the pellet so hard because they don't have a dust. That's the problem. So the chick going to refuse to eat the pellet one. Oh, you can see this picture. There's a lot of feed waste here because the chick just bite the pellet feed and then just throw away like this. So that's going to affect the feed intake a lot. For layer, very similar to the broiler. During the past 50 years, a lot of improvement. And right now, we go to the genetic that can lay 500 eggs in 100 weeks. So everyone happy about that, right? Because you can get a lot of eggs from that. But there's a lot of things that you don't understand what's going to happen with that kind of production. You're going to have a less egg weight. I think in Thailand we have a problem with 1 or 2 grams of less egg weight compared to in the past. And then we have a problem with the body weight of spent hens. Thailand is not like in US, so we can sell the spent hen to make food, and it's a very good price. I think Philippines or Malaysia may be the same. So it's very concerned because if you have a genetic that lay a lot of eggs, you need a bigger size of the chicken, right? You need a small frame. So that's the real problem. And another one is the egg and eggshell quality. because we get confused how to feed them correctly. This is the typical graph that shows the performance of the new genetic of layers. So you can see that for the egg production, this is the previous genetic, the dot line here. And you can see the egg production of the new genetic. If we counting the laying hens at around 80 weeks of age, some farms they can produce more than 80% of eggs. And we have a problem with the egg weight here. You can see it's about 1 to 2 or 2 to 3 grams less our egg weight. And body weight at the end, that's going to be the problem that I already told you. So this is the things that happen nowadays, especially in the tropical conditions. So what we should do about the nutrition, normally we have a diet during the laying period, like a layer 1 and layer 2. Layer 1 is a high in protein and metabolizable energy, but low in calcium. But for a layer 2, maybe a little bit less in protein and energy, but higher in calcium. So normally we change to the layer 2 at around 50 weeks of age, because we thought that at that time the egg production going to be down right here. So it's time to change the diet. But you can see that the eggs, the chicken still lay egg at this level. So you change to the lower nutrient density of the diet. So it's going to have a problem with the layer. So most of the farmers right now, they move to change from the layer 1 to layer 2 around 70 weeks of age. But what's going to happen when you change the diet to layer 2 at 70 weeks of age? The calcium level is not good enough for the bone of the layer hens and the eggshell quality. So you need to supplement with like a oyster shell. So that's the another problem. But right now we know how to handle for the protein and the energy in the diet for this kind of genetics. It's very funny for the Thai industry, because we have an egg board. And the egg board, Thai egg board, try to control the number of the laying hens, around 50 million birds. Then they expect that we're going to have like 41 to 42 million eggs per day. That's going to keep the balance between demand and supply. Right? So they start counting the laying hen at 78 to 80 weeks of age. So that's, I told you that at that time, the hens still lay more than 80% of egg. But we still have a problem with the body weight, the less body weight of the spent hen at that time. So after I have an idea, I talked with C. Wiley about the celluTEIN product. So I asked a sample and then try to make a little experiment. Try to increase the body weight during the last month of production. So we use 180, 87 week of age high-sex browse. And we have a two treatment with and without celluTEIN supplementation. We use the level of 66 gram per ton for 4 weeks. And we have a 6 replicate with a 15 hen each, and we conduct this experiment at our facility at Kasetsart University. The production is not different at all. No different in production. But what we've seen is very interesting. This is before, this is the control one before we start the experiment. We look at the albumin height here. And this is the celluTEIN group right here. This is before the experiment start. And then after 4 weeks, we measure again, and you can see that we have a drop in the albumin high in the control group, but for the celluTEIN group, we still maintain the albumin high. It means that we can maintain the egg quality during that time. And we look at the whole unit. I think it's the same. So this is the beginning of our observation. So we know that for this kind of experiment, it's not like a professional one because it's really, we have just an idea and try to observe what's going to happen. So right now we conduct a trial from the beginning when we start, when the chickens start laying eggs. So what's going to happen, I think if I have time or we can meet again in the future, I'm going to show you that result. But this one, I think very interesting because we've seen the improvement in the egg quality when we're using the celluTEIN products. So for the summary of this experiment, the supplementation of celluTEIN at the last month of laying period, as I told you, more than 80 weeks of age, did not improve the body weight of the spent hen and egg production, but it's improved in egg quality in terms of albumin high and whole unit. For pigs, I think right now everyone knows that we have a very good genetic for the litter size. So right now the sow can give birth. You can see, you can count, I think more than 20, maybe 24, 25 piglets. And they have only 14 nipples. So the big problem of the modern pig breeding right now, we have a less average body weight of the piglet at birth, and large number of piglet born less than 1 kg. High mortality prior weaning, and the high mortality and morbidity after weaning. So this is a big problem nowadays in our pig industry. So you can see that when the litter size increased, this is the number of total piglet born, and you can see the average birth weight is declined. And right now, I think for the total piglet born, right now in Thailand, it's allowed this number, 13 to 15, and you can see the average of birth weight is less than 1.5 kg. And when you see, look at the distribution of the piglet weight individually, you can see a high number of the piglet born that less than 1.1 kg or 1 kg. You can see higher and higher of this piglet in your litter. So how we can improve this problem? The consequence of the small piglet born is we're going to have a wean small size piglet more and more like this. In terms of the gestation physiology, we know that the development of the piglet during the gestation, the placenta, the udder, always occur during the last third part of the 1/3 of the gestation period. So the key is not to feed the sow too much during the first of the gestation period. So that's the key. So I think we recommend the farmers try to not have like a body score of the sow at 12 weeks of gestation more than 2.75. And this picture compare the sow that have a body score more than 3 and 2.75. If you feed the sow and let them have a body score more than 3 at 12 weeks of gestation, you're going to have a problem with you cannot feed enough or more than cannot feed a lot of feed to the sow, because you're afraid that they're going to be bigger and bigger and cannot give birth very well. So we recommend the farmer to have like a body score at 2.75. And during the last 1 month, we can increase the feed intake of the sow. And then have a sow at the farrowing about the body score about 3 to 3.25. So it's this picture like this, not this one. And that we can have a very good in uniformity of the piglet born right here. And the average birth weight going to be more than 1.5. And you can see a good development of the udder also. So this is the management way to work with a genetic that have a higher litter size right now. But if we can have like another solution, I think it should be great. Another thing, maybe it's going to work very well also when you have conduct an experiment with the weaned piglet. We recommend the farmers to make a group of the piglet when we wean the pig. And in this case, we wean at 3 weeks of age. So we separate the piglet into 5 groups. The grade A1 is more than 8 kg. The grade B, 7 to 8 kg. C, 6 to 7. D, 5 to 6. E is less than 5 kg. And then we list them separately. And look what happened if we grade grading the piglet like this. We can see normally we expect the normal distribution. But if you have a very good management, you can have a curve like this, not a normal distribution. And if we see this kind of picture, what kind of piglet is going to have a problem with the growth and development after weaning? It's going to be grade D and E. So every farm can know, okay, how many piglet that wean in these categories, and then we have need a intensive care unit for this kind of piglet. But if the farm have a good management, there's a less pig in this category. And what we do, we have a very intensive care unit. We have a good brooder, we have a good feeders, water, and that can help a lot. So this is one for the grade D and E piglet. So I recommend you, if you want to study about the effect of something, the piglet after weaning, try this group of piglet. Because if you use the average one, sometimes you cannot see the difference. But if you just use only the small one, maybe you can find something very interesting. Another problem with the weaning process is the post-weaning diarrhea. And that because of the stress during the weaning period, change in environment, the gut intestine development and challenges, not good enough, social changes, and then you have a pro-inflammatory. And then after that, if the situation gets worse, you got infected by bacteria, and then you have a diarrhea. So this happened. So I think many products right now in Thailand that they try to stop the post-weaning diarrhea. And this product is in the category of the anti-inflammatory compounds. We have antibiotic growth promoters, but right now it's already banned in Thailand. So many people looking for the NSAID, using aspirin or ibuprofen. Phytochemical compounds and the postbiotics. So I think this is going to be very interesting in the future, that how we can correct the problem of the post-weaning area. But I would like to emphasize that the management is the key one. If you try to reduce the stress to the animal, it's going to help a lot. So this is the summary of the critical problems in today's livestock and poultry production. For broiler, the gut, immune, and body development during the first 7 to 10 days, how we can enhance them. The protein nutrition, the feeding management during the 7 to 21 days of age. For layer, how to collect the less egg weight. Less body weight of the spent hen, and the egg and eggshell quality. For pig, less average body weight at birth and high number of piglet born, less than 1 kg, and high mortality and morbidity prior and after weaning, and the gut inflammation during the weaning period. So I think I'm going to give this question to Brad and CY to collect. It's going to help our livestock industry in Thailand a lot if you can can correct this kind of problem. So I would like to summarize my talk that this kind of problem, I think it's very important that you have a good nutrition and management strategies. How we can provide the complete and balanced nutrient intake and utilization. Not just the qualitative one, but quantitative also. How to enhance the animal to eat more and more. If you can do so, you can enhance the gut development, preventing the gut inflammation, and growth and development, and enhancing immunity. And very important, our production right now, actually there are 2 factors. The one that within our control, the genetic, nutrition, environment, management, the husbandry, and the health, the disease. we have a knowledge about this. So this is within our control. But we still have another factors that without our control. There's a lot of situation in the world that going to affect our production. So we need a real-time nutrition and management, not try to find the absolute solution to correct the problem in any situation. So we have to go one by one, and then using our basic knowledge, and then our production. So I think this is going to be the key to correct the problem that I already told you. So thank you very much. So if you have a question, I'd love to entertain your question. We failed to demonstrate that, but I think because we have a very short time for preparation, we just got an idea, and then we try to find the laying, and then we apply. So right now, I think if we have to do that, we have to do at the start, from the beginning, and look what happened. So right now we conduct that kind of experiment. Maybe you need another 10 months to have a good result for that.
Good afternoon. For those of you I haven't met, my name is Brett Casebolt. I'm the co-founder and CEO of celluTEIN Bioscience. I'm joined here today by my fellow co-founder, Matt Jones. We also have Melissa Werner here, who's actually put this whole program on, so I'd appreciate maybe a little round of applause for Melissa. And our Director of Asia Business Sales, Mr. C.Y. Lee. Before I begin, I have to say a sincere gratitude and thank you to the professors. Despite we may have heard they weren't paid, so they've actually chosen to do work on this product oftentimes on their own. We've talked about ideas, they want to go certain ways, and as you'll see when I take you through a very high-level overview of our data and information, we're a very data-centric company. There's not a lot of fluff in our stuff, so it's a very heavy on the data, heavy on the technology, and you just have to tolerate me for a little bit. So I apologize for that. But should you have more information and want more details beyond what we're going to show today, because we're just going to show a high-level overview of the celluTEIN product, please contact Mr. C.Y. Lee, and he can certainly get you what you may require and give you more information details. So by way of background, we started this company about 15 years ago, and it's kind of interesting to me to look around this room now here today and see people that, we've met since the very beginning, and some people we met along the way, and people who we're just meeting now, and it's very exciting to see how far we've come. Our internal team, outside of us business commercial operators, is comprised of experts in protein chemistry, muscle biology, as well as immunology. Our company is PharmaQS, GMP Plus, and HACCP certified. We're here to talk about celluTEIN. You've heard a lot about it, and so I'll give you a little bit more of an overview on the product. I'm getting the signal that I'm not speaking the microphone, so. celluTEIN is a yeast-based Saccharomyces cerevisiae product, and that is the basis of the product, but that's not at all what the product is, and I'll describe in more detail what that is. It contains a high level of functional proteins, metabolites, amino acids, things of that nature. And it's been demonstrated and proven to stimulate the mTOR pathway. And key in this is we have a proprietary biochemical process we developed many years ago that not only increases the bioavailability of these key proteins and delivers them to the animal at the appropriate position, specifically like the upper digestive tract, but also stabilizes these products through the rigors of modern production. So we've got tests in autoclaves, we've got tests in extrusion, and with very high, nearly 100% survivability. We've proven our product in a wide range of both vertebrate and invertebrate animals, so it works across a wide range of species, everything from Pacific white shrimp to monogastric swine. and birds, et cetera. And the product's currently registered, and I may have some of this wrong, so you have to check with CY on this. United States, Mexico, Philippines, Korea, Thailand, Vietnam, India, as you mentioned, doesn't require it, Malaysia, and China. And we've completed numerous university trials, as well as we don't have any internal research facilities. We don't even have an animal nutritionist on staff. So all of our work is done at third-party pharmaceutical-grade contract research sites or universities. So again, kind of back to the point of we're a highly data-centric company. And we've done work in nursery pigs, sows, grow-finished pigs, broilers, layers, turkeys, not a lot of those over here, pullets, sea bass, Asian sea bass, and Pacific white shrimp. So you've heard a lot about mTOR, and I'll talk a little bit more about it, not to completely beat you up about it, but it's the nutritional link between health and cellular growth. So when we started this company, started developing this product, really the scientific side of our group said, Dr. Ron Strobing, who was the founder of the technology, said, we really need to break this energy balance, this diagram that Professor Gonzalez showed us, where when animals are challenged or under stress, less energy can go towards growth. And said, so we set out with these questions, including how do we break this energy balance during challenges to encourage more of the feed energy or caloric energy to go towards growth? Also wondering, how can we stimulate an animal's own immune system? So there's lots of products out there that Professor Sexton talked about, antibiotics, NSAIDs, things like that. There's a lot of very specific therapies, antibiotics, vaccines. But our thought was there's no better immune system than Mother Nature. So if we can figure out a way to stimulate an animal's own immune system, specifically their innate as well as adaptive immune system, perhaps we can get a little better response. And how do we do this and get an atypical cellular-based musculoskeletal growth? And how do we do all of this through nutritional additives? And we found our answer in mTOR. It is the crossover of health and nutrition, and growth and immunity are linked by mTOR. I won't belabor because this is an eyesore right here, but basically this is demonstrating both the metabolic function of mTOR metabolism across a variety of tissues, and this is showing actually the opposite, that inhibition of mTOR prevents immunoglobulin development. So just a little bit more on mTOR. So you'll hear it called also mammalian targeted rapamycin, but broadly speaking, and in birds, they'll more generically call it mechanistic targeted rapamycin. It's highly conserved across a wide variety of species, everything from not only mammals and birds, but also to plants and even yeast. In fact, I'll come back to that in a minute, but mTOR controls cell growth, and as I mentioned, is across our path of health and nutrition, and was originally discovered in 1972 on Rapa Nui or Easter Island, and that's hence where rapamycin name came from. And so what they did was they discovered a bacteria they extracted out of a Saccharomyces yeast. And that bacteria turned out to be was able to be isolated and found out to be rapamycin. And what rapamycin does is rapamycin actually represses the immune system. So it actually brings the immune system down by inhibiting T cell and B cell activity. And so today, even, rapamycin is broadly used in human use in the context of organ transplants to suppress the immune system. So when you have an organ transplant, you don't reject it. So we thought, well, what if we could cause the opposite effect by stimulating mTOR? We might have an effect that would stimulate B cell, T cell, as well as perhaps some other immune responses. And what we have demonstrated and proven, and again, like I said, there's tons of detail behind all this, so I apologize. I'm used to my standard pitch where I go into proving to you that we stimulate mTOR, so I apologize. My flow's off a little here. But we have proven we stimulate mTOR and create this atypical response using these functional proteins, which consist of peptides, amino acids, nucleotides, found within these yeast, as well as the additions of certain key amino acids. And what we do is we then process it using our proprietary biochemical process that increases the effectiveness of this and has been demonstrated to be stable over a wide range of pH and at very high temperature ranges and pressure. And it's effective in a wide range of animals. So we can take these functional proteins, we can process them using our biochemical process, and then ultimately trigger the mTOR effect, which yields, stimulates the animal's immune system as well as leads to musculoskeletal development at a cellular level. Here's the proof I was looking for. So how do we know this works, and how do we know that that celluTEIN stimulates mTOR? This was work originally done at the University of Minnesota Veterinary School with Dr. Michael Murtaugh, who was a PRS expert and did a lot of consulting work for Boehringer Ingelheim. And his postdoc student, Dr. Michael Rahe, who's now at Iowa State University, helped us follow up with some of the studies in celluTEIN. And what they did is they took porcine B cells, and then this is on a piece of equipment called an Agilent seahorse equipment. And what it does is, instead of your typical assay equipment where it measures a point in time, it measures the same sample multiple periods of time over time. So you can see response by a time curve. So what this is doing is it's looking at the effects both aerobically and anaerobically over time. And porcine B cells, the known control to stimulate effect is CD40 plus interleukin 21, is known to increase the activation and proliferation of porcine B cells. So you see that right here. You see a nice response to it. And then what we found was by dosing celluTEIN, we got a slightly better response numerically and statistically. However, when you take celluTEIN plus rapamycin, which is what this one is, you lost all the effect. So again, we saw we were increasing, and there's a lot more data behind this and it's been published out there, but we were increasing the energetics and proliferation of porcine B cells. So we're showing an increase of B cell activity, but all that effect was removed when you put rapamycin on it, which the only known mode of action for rapamycin is to inhibit mTOR. So the only way we're stimulating the B cell immune system response is through the mTOR pathway. I'm going to give you some overview on a variety of our trials, like a one-page summary. There's, again, much more data behind this. So I'll go through poultry, swine, and aquaculture for you. This was the first broiler study we did. This was done at a research facility on the eastern seaboard of the United States. This group does contract research for a lot of the big major drug companies found in the United States. And this work was also co-authored by Dr. Park Waldrop, which you saw Professor Sexton show a moment ago. So celluTEIN was fed at 300 grams per ton in the starter phase and 50 grams per ton in the grower and finisher. And they did a clean, a low stress or clean conditions, as well as a high stress or challenge. The challenge, they used dirty commercial litter, found at a commercial operation with challenges, as well as added coccidiosis and clostridia to the litter. So you can see at day 42, you can see the negative effect, almost a loss of 10% on the negative control when challenged. Here you see, and so what this would be is, this is, as I mentioned, a contract research organization, does work for a lot of the drug companies and as well as some of the antibiotic companies. So this is a typical antibiotic setup. So what the goal is you show the clean control, then you show the challenge and the poor performance in both weight growth as well as feed conversion. And then the goal is, under this situation, to use your treatment to try to get back in line with your control. So, which was achieved on a feed conversion. We saw an increase in the challenge of about 11 points of feed conversion, and statistically, numerically we were better, but statistically the same. Numerically and statistically, we actually got more gain than the original control, so actually outperformed it. So again, under the low stress condition, we saw about 7 points of feed conversion improvement, and under the challenge, about 13 points, as well as increased uniformity. This is a summary of an egg layer trial we did. Again, this was, as Dr. Sexton said, he was doing just kind of a very end, feeding at 66 grams per ton. In this trial, we fed 66 grams per ton for apologize, I got to think of this off the top of my head. I want to say it was for 20 weeks. Is that? I could be wrong on that. It might have been. I apologize, but again, we've got backup on this. It was fed for a much longer period of time. We used standard diets versus 3% lower amino acid diets, so to kind of more mimic people that are limiting amino acids. Crude protein and metabolic energy were maintained. We saw the following. We saw an increase in hens active in production, increased from 77.6 to 85.3%. We increased average egg weight, egg thickness improved, and feed conversion improved when kilograms of feed to kilograms of eggs. So, and you can see the data here, both under the standard diet as well as the lower amino acid diet. This was an in vitro study that was recently done at the University of Minnesota Veterinary School, done by a virologist by the name of Dr. Sagar Goyal. Dr. Goyal looked in vitro using H4N6, low pathogenic avian influenza. And what he would do is he would take these MDCK cell monolayers and he'd either pre-treat them with celluTEIN and then incubate them and then add the virus, or he would add the virus and the celluTEIN at the same time, or he would add the virus, incubate the cells, and then add the celluTEIN. And then they measured the response in log reduction. So this would typically, you could think, this really is designed as more of an antiviral look. So typically when you see the antiviral products, you would see a really good response here because the products are pre-coated on the cells and then the virus comes in and doesn't work. We don't typically see a lot. We get better response further out. Again, hard to interpret what that means, but if you look at some of the data we've done in the past in live animal trial work and challenge trial we've done, it would show more of an effective response by stimulating immune system responses in the animals is the best way I'd say characterize this to in vitro. But a very nice, and I would say he said, very sizable reduction in virus counts in that study. Regarding our swine trials, in this nursery study, we fed 175 grams per ton for days 0 to 42 post-weaning, and we added about 3.6 kilograms at the end of the nursery. Pretty significant gain. We also improved feed conversion by 6.8% or 15 points. Furthermore, we actually added an additional 1.1 kg in the next 14 weeks, so even post-feeding. So very, very good response. I wouldn't say I would expect that consistently. This was a swine nursery and grow finish study we looked at. So here we either fed 175 grams during the entire nursery, days 0 to 42, or we fed 100 grams per ton during the entire grow finish study. So either nursery, finish, grow finish, nursery, grow finish. So what you see here is at day 42, this one was more like 1.4 kg gain, feeding it in the nursery, more I'd say typical. However, just by feeding it in the nursery, ended up adding over 4 kg at finish. We also saw a nice improvement actually even in the grow finish feed conversion ratio just by feeding it in the nursery phase. And nice improvements in fecal conversions. In the grow finish side, we saw about a 14-point improvement in feed conversion just by feeding it in the grow finish. This is work we're continuing to do more on, and we're going to do more scale work in this. There seems to be a lot of interest in grow finish pigs and trying to figure out a way to pick up that kind of lost effect of the removal of ractopamine pretty much across the world. So. Dr. Sexum talked a bit about, how do we help these young pigs, these baby pigs, and I'm a big believer that it's in the sows. It's in the sow diets. And I think if you think back to what Dr. Gonzalez had presented and talked about as far as timing of developing muscle development and stuff like that in utero and during gestation, I believe generally speaking, we've got very good, complex nursery diets today. The sow diets are are very basic by comparison, and I think there's a lot more that can be done in that area to improve performance in the offspring of sows. The genetics have done a great job, but I don't feel like the nutritional side is kept up as well there. And I get it, it's a cost thing. So in this study, we fed sows either 175 grams per ton or 500 grams per ton, which is what we were dosing a previous product of ours at. And feeding 175 grams per ton with a p-value less than 0.05, of the pigs born of the sows fed the product, we increased market weight of those pigs born on average of 3.3 kg and had an improvement of feed conversion of 10 points. And I don't know why that, yeah. And we also improved wean to estrus interval by 1.5 days. And that's consistent with our previous work where we've seen anywhere from 1 to 1.5 days pretty consistently. This is some work that Dr. Matt Vaughn did for us internally, taking a look. You may recall Dr. Gonzalez talked about the 16% increase in protein synthesis in neonatal, but we also saw a nice increase in fetal myoblast cell proliferation. So demonstrating that we're able to show that celluTEIN has two modes of action. It works to not only increase the size of muscle cells, but also increases the number of them. Again, going back to Dr. Gonzalez's talk, if you can feed them early enough in the sow. We did 2 studies here at KU with Professor Orapint, and the first was in Pacific white shrimp, and she fed celluTEIN at either 100, 300, or 500 grams per ton, and then she did it both with and without an EMS challenge. Specifically, this was a low pathogenic dosage of Vibrio, and we found the following statistically significant. Well, first, in the challenge study, the negative control had about a 23% mortality, and while not statistically significant, even at 100 grams per ton, we cut 10% of that mortality away, and at 300 grams, we got a statistical significant difference, and obviously continued to numerically decline as dosage went up. We saw a statistical improvement in FCR of 5 to 7 points when it was unchallenged, and increased weight at 8 weeks of these shrimp by anywhere from 5.5% to 15% over negative control. We also, in the challenge, we reduced Vibrio counts within the hepapancreatic sac of up to 1.5 log reduction. So if you remember some of the in vitro data I was showing on AI, here again we're seeing, you know, a Vibrio count reduction, as well as an increase in some innate immune system biomarkers, in this case lysozyme and phenyloxidase activity. In the Asian sea bass study that she did, here she did a more linear response with a more linear treatment, 150, 300, or 450 grams, and then either challenged them with Vibrio or with Aromatis. And at 8 weeks, this is the results of the FCR at 8 weeks on the unchallenged portion. So very nice statistical response as well as numerical response over the dosages. Within the Vibrio challenge, we saw a nice increase of in the acquired immunity response of IgM, as well as the Vibrio count in the blood reduced at about 300, started reducing statistically significantly, but a nice numerical linear response. In the Aromatis, we also saw a nice statistical response and growth in immunoglobulin M, as well as a reduction in the Aromatis counts. So in summary, we have the real key secret of what we've got. There's nothing really overtly magical about what we do or the composition of our product, but what it is we really have is this biochemical process that we use that not only increases the stability of the celluTEIN product, but actually also increases the bioavailability of the key components within the delivery of it. We've demonstrated it stimulates the mTOR pathway and across a wide range of species, both physiologically and immunologically. And so when we go back to those questions we tried to address at the very beginning, we have demonstrated that celluTEIN breaks the energy balance problem during challenges. We've been able to demonstrate we improve FCR in a variety of animals during challenge studies. We've shown we stimulate the animal's own immune system. We've increased innate immune system response as well as adaptive immune response during both viral and bacterial challenges. And we've created an atypical or almost pharmaceutical-like cellular-based musculoskeletal growth. We showed in vitro how we have improved musculoskeletal growth, both in the number as well as the size. We've demonstrated in with the work done by Professor Kim, we've demonstrated in swine that we've increased villi height and crypt depth. We've also, in our own work, we've seen the same results in broilers as well as pullets. We've increased villi height and crypt depth. And we've also seen an improved reproduction response in egg-laying hens as well as pigs born from sows fed. So in summary, celluTEIN is a proven bioactive nutritional feed additive to meet the modern needs of animal production. So that's the summary. Any questions? Yes.
How do you differentiate your product with nitric oxide or IgE?
Right. I would say performance at the end of the day. It's a good question. I'd say at the end of the day it's performance. Actually, I think, did you do some work on nitric oxide or no? For some reason I was thinking you did. Yeah, I think there's a lot of products that can target mTOR, as we've seen, a lot of different things out there. And we've looked at a lot of these products as well. So there's things, including drugs. And what we've generally demonstrated and what we've seen, at least in vitro, as well as what we've seen as far as live animal studies, the performance of our product seems to be much better. The arginine, in order to get the same arginine response of what we've got, so we did about a 5,000 bird broiler study, 42-day broiler study, and we had arginine, leucine, and glutamine, and we had it with and without in our product, and we added it at 5 grams, all of those combined at 5 grams per metric ton. And over 42 days, we got 2.8 points of feed conversion, and if I remember correctly, I think like a 3% improvement in overall growth. So adding 5 grams per ton, I mean, an inconsequential amount. There's a couple of studies out there about, I know using glutamine anyway, where they had to add I can't remember, it was like maybe, I want to say, I can look it up and get it to you here before we're done with dinner, but I wanted to say it was like 20 times or 25 times that amount to get nearly the same response.
Will it have a complementary effect with the
We have, it's a great, that's why we did the amino acid studies. So in that egg layer study, you saw we reduced the amino acids, and we did a similar study in pullets as well, which I didn't show, but it's same thing. We were trying to see how much of an impact just the amino acids would have. We've certainly seen it still works at lower levels of amino acid, but in my opinion, which is just an opinion, I think it works better and should be more synergistic, because when you stimulate that mTOR, and there's experts in this room who are better at answering this than me, but my understanding is when you stimulate that mTOR pathway, you're going to stimulate more of a nutrient uptake. So one of the areas, like I reference ractopamine a lot, because in swine that was a big loss, but you think of that one, it required, and in order for it to work effectively, you needed to add more lysine to the diet to make it work. So that's an area that I would say we have of interest, and we want to explore more, is can we get, how much more synergistic can it work? But yes, it scientifically should work that way, and we've demonstrated when we do the higher like 3% higher amino acids with same metabolic energy and crude protein, we get a better response. So it should be, it'll be more synergistic with that. Any more questions? No. Sorry? Sorry? We've titrated a lot, and our recommendation is 300 grams per ton in the starter, followed by 50 grams in the grower and finisher. Now, there's some issues with that, because as you all know, when you start getting down to 50 grams, a lot of feed mills will say, sure, we can do it, but we've demonstrated that's very hard to do, so we're actually currently working on a diluted product that would be, or dose it at 10x that level, so you can ensure even distribution. Because, you know, we've met with the biggest operators in the world, and they all kind of say, when they're being honest, they start questioning how they're going to do it and whether they can do it and stuff, and then they all say, oh yeah, we can do it. So. Well, based in broilers, we haven't tested it in broilers, but in pullets and layers, it looked like we could. I'd say, and this is a point that I think if I recall correctly, Professor Sexton had brought up, is he was wondering, I think, and correct me if I'm wrong, was that maybe rather than even looking at that, as long as you're above the minimum legal threshold, whether you can reduce the crude protein perhaps. Again, kind of getting more nutrient uptake by, you know, if you can reduce some of that, you might get it. So you should, in theory, you know, be able to reduce the amino acids and get the same response to the control, but it depends on how much, if there's excess amino acids or not. Very good. Okay. Well, again, thank you all, and appreciate everybody coming out tonight. And we have a wonderful dinner and some gifts, I think. So please, we're hopeful you're able to stay around and enjoy a cocktail hour and some live entertainment.